students. Today we are going to discuss numerical chromosomal aberrations, and this is study material for 4th semester student. As we know that in somatic cells of every organism there is a specific number of chromosomes which are found in pairs, and at the time of gamete formation due to reduction division, chromosome number is reduced to half. So the gametes carry haploid set of chromosome and it is represented by X. When we talk about the alterations or aberration, or you can say variation, that is in terms of numerical chromosomal aberration, it is variation in the number of chromosome from the diploid set. It is of two types, euploidy and aneuploidy. It can be better understand by the flow chart. So we will see here, we can see here this chart, that ploidy is of two types, euploidy and aneuploidy. Euploidy means that the organism should possess one or more full set of chromosomes. It is of three types, monoploidy, diploidy and polyploidy. In monoploidy, there is a single basic set of chromosome that is represented by N, and in diploidy, there is two haploid chromosome sets, and in case of polyploidy, more than two haploid sets of chromosome are there. It can be of two type, autopolyploidy and allopolyploidy. As the term indicate auto means self. When the chromosome set in an individual belongs to same species, it will be result in autopolyploidy, and when the chromosome set in an individual belongs to different species, it is called allopolyploidy, as allo means different. Allopolyploidy can be explained with the help of example of triticale, which is first man-made cereal. Here two different species are crossed. First plant is Triticum durum and second plant is sickle cereal. In Triticum durum it is tetraploid plant, and in sickle cereal it is diploid plant, and when the cross is made between Triticum durum and sickle cereal, F1 hybrid is produced, which is triploid 3x is equal to 21. As in case of Triticum durum, after gamete formation there will produce 14 number of X, and here in case of sickle cereal it will produce X number 7. So 21 14 and 7 number is equal to 21. So here hybrid which is produced, it will have diploid set having 21 number of chromosomes, and in terms of ploidy it is triploid and their number is 21. After doubling with colchicine treatment, chromosome number become doubled and it become hexaploid, 6x equal to 42. It is an example of allopolyploidy. In case of aneuploidy, as we discussed before, in case of aneuploidy, it means presence of chromosome number which is different than a multiple of basic chromosome number. It is of two type, hypopolyploidy and hyperpolyploidy. In case of hypopolyploidy, hypo means anything is lost and hyper means anything is gained. In case of hypoploidy, loss of one or more chromosome in the basic set of chromosome and basic diploid set of chromosome, and in case of hyperploidy there is a gain of one or more chromosome to diploid set of chromosome. Hypopolyploidy is of two type, monosomy and nullisomy. When there is a loss of one chromosome from diploid cell, it is represented by 2n-1 and it is called monosomy, and in case of nullisomy there is a loss of pair of homologous chromosome and it is 2n-2. represented by twin minus 2. In case of hyperpolyploidy, trisomy and tetrasomy. In trisomy, there is addition of one chromosome to the diploid set of chromosome, and in case of tetrasomy, there is addition of two chromosome to the diploid set of chromosome. So we have discussed here about euploidy and aneuploidy. Now we will discuss about the significance of this ploidy, how it is important. It is very important. It play important role in plant breeding and horticulture. And ploidy crops are large in size. They give increase in yield and they produce large flowers and fruits. Hence, they are economically very significant and they play very significant role in evolution of new species also. And due to these commercial importance, they are this topics is important for study point of view. So we have discussed today here numerical chromosomal aberrations. Thank you.